Good morning. Thank you for making time this morning. My name is Mindy Oh. Today, I would like to introduce our core technologies, our pluripotent stem cells, newly elicited pluripotent stem cells, and EPS. These are topics we are going to cover today: company profile, treatment case, and EPS, Parkinson's disease treatment case, and self-differentiation from NEPS. Since 1989, for almost 30 years, we have researched on stem cells, gene, and new drug development. And we successfully differentiated stem cell into human pancreatic beta cells, hepatocyte, neurocyte, chondrocyte, osteoblast, and kidney cells. STRI has applied 34 patents over 146 countries related with NEPS, and 16 patents are registered in Korea. STRI is the cooperation to develop the human tissues and organs. And also, STRI was first developed the pluripotent stem cells without side effect. Among various treatment cases, today I would like to introduce alopecia universalis. Most cases of alopecia universalis occurs before the age of 30s. In our case, early 20s, the female, the patient. Alopecia universalis is one of autoimmune disease, losing the hair on own bodies. This slide shows we use mesenchymal stem cells for treatment from umbilical cord. As you can see, figure A is before the treatment, and figure B is after 12 times treatment through local injections, and figure E is after 20 times treatment through local injection. Each time we are injected 10 million cells through local injections. STRI has accumulated many mesenchymal stem cell treatment research, and based on those research on mesenchymal stem cells, STRI has developed pluripotent stem cells. NEPS has unique characteristics. It is for immediate human use. It has no side effect, and it is mass producible. If 1,000 fibroblasts are induced for differentiation, less than 100 IPS produced. However, example, we induce 1,000 MSC into over 900 pluripotent stem cells without side effect, and this means we have production rate over 90% success rate. We use mesenchymal stem cell to induce pluripotency only using natural compound. And this natural compound is originated from plant. And we applied patent with this natural compound over 56 countries, and which is called STC-nEPS. This slide shows NEPS is originated from mesenchymal stem cells. We use pluripotent stem cell marker to identify whether NEPS is pluripotent stem cells. We analyze the level of 4 master gene expression: SOX2 nanook, OCT4, and CMYC. As you know, stem cells master genes SOX2 nanook are increased in gene expressions. Moreover, ACT4, which play a major role for pluripotent stem cell, is increased. However, the CMYC, the cancer expression factor, has reduced than MSC. Therefore, DNA analysis confirms nEPS is pluripotent stem cells. This slide shows the in vivo experiment tested cancer expression in mice without immune system after nEPS injection to seminal vesicle. As a result, iPS expressed cancer within 4 to 6 weeks. However, nEPS did not express cancer even after 20 weeks. This slide shows biopsy photos of mice seminal vesicle. We have done tissue analysis and proved that there are no tumor occurrence. And we have lots of experience in Parkinson and Alzheimer's disease treatment. I would like to show the one videos. The patient who had Parkinson and Alzheimer's disease for 10 years. The first video shows this is before the treatment. He can hardly move himself or open his eyes. The second video shows the patient after 3 times stem cell injection through IV. We injected 60 million cells per day for the first 3 days. After 3 months, we repeated treatment. And the last video shows the patient with total 7 times injection through IV. He has significantly improved motion and cognitive abilities. We are still following with him, and this video is we are started the injection this 2014, 2015, so we are still following with him, and he is maintained the symptom at this time. Okay. And I would like to introduce the derived cells from nEPS. We are, as I mentioned, we are successfully differentiated stem cell into pancreatic beta cells, hepatocyte, neurocyte, chondrocyte, osteoblast, and kidney cells. The first one is pancreatic beta cells. This slide shows this experiment regarding pancreatic beta cells differentiation from nEPS. The 5-lane demonstrates that insulin gene is expressed. It is pancreatic beta cells in glucose control functions. nEPS and beta cell differentiated from nEPS injected group showed a drop in glucose level after 7 days of injections. It is a comparative study on insulin production among iPS, human healthy pancreatic beta cells, and NEPS. NEPS differentiated beta cells released higher insulin levels than human healthy pancreatic beta cells. And we are collaborated with all national universities, type 1 and type 2 diabetes animal modeling test. It is a beta cell glucose level regulation in type 2 diabetes. Beta cell differentiated from NEPS have decreased the glucose level in type 2 diabetes modeling test. Insulin injection is a general treatment for diabetes. However, this method is problematic since injected beta cells are dispersed throughout the body. And also, we are considered as immune rejection and longevities. So STRI designed a pouch to hold insulin at certain locations. Pancreatic beta cells secrete insulin inside the pouch. However, the pouch's pores are small enough that only insulin can penetrate the pouch. The pouch is designed through 3D bioprinter with PLA. This is the pouch. This slide shows, using this pouch, we conduct type 1 diabetes animal modeling test. The result is as shown. Our glucose level of cell pouch has reduced as much as 40% compared to control group. Red one is control, and the cell pouch is blue. So the research focus on iPS is mainly to overcome side effects, and they spend enormous research on these issues. However, we eliminated all side effects and maximized the production efficiency through our NEPS. And also NEPS can be commercialized immediately, and it will be shipped the paradigm in biopharmaceutical industries. And NEPS is opening to new chapter to cure the incurable disease. So we are introduced our core technologies, pluripotent stem cells today. Thank you for attention. Thank you.